This is the last part of the content related to the genetic inheritance. And we're going to talk about behavioral genetics and epigenetics here. And I'm thinking this will be 10 to 15 minutes long. So the word heritability just refers to the extent to which a trait is passed along from one generation to the next. And so we can look at this example of height here, and we can see that in this there's a lot of variation in height within this group, and that some of that is genetic, some of it's due to the environment in the model that's present for this part of the lecture. And when we look at how much of height is due to genetics, we'll look at how we do that in a moment, but we would say then that if a lot of it is due to genes, as in this pink bar here, that the trait is pretty heritable. If we look at another trait, like the likelihood of napping, we find out that the genetic contribution to it is really small, as you can see on the graph on the right, but that habits or the environment, like having to go to work early or noise pollution or technology distractions, all relate to the necessity of naps, so there's a much greater control of the environment. And so we're always looking at, when we have a trait, how much of it is due to genetic inheritance. And how can we know? So the way that we can tell the heritability of a trait is often done through concordance studies, and most particularly twin concordance studies. Twins are particularly important, and especially monozygotic twins, one zygote, one fertilized egg twins. You can see that we have one egg, one sperm that's separated into two separate beings along the way, meaning that these two babies share all of their genes. We can then tell, to some degree, how much the genes impact the outcome. On the other hand, dizygotic DZ twins, often called fraternal twins, no more similar than any two siblings. It's one egg from mom, one sperm from dad, a totally different egg from mom, a totally different sperm from dad. They will share about half their genes. They might be the same sex or different sex. They're no more similar than you and any other non-twin sibling. And when we want to look at how similar they are, we look at how much they resemble each other on certain traits. So we'll take the trait reading ability, and we'll plot the reading ability for the firstborn on the scale from 0 to 100, and we'll give the same test to the secondborn on the scale of 0 to 100, and we'll plot a point. The point for the secondborn, this particular lower spot right here where the arrow is, that secondborn got a 60 and the firstborn only got a 10. But in most cases, you see that if the firstborn was higher, like most of them, the firstborns got 80, the secondborns were higher. And we can see when we look at correlation scatter plots that a nice tight plot like this means there's a pretty strong relationship between reading ability between firstborn and secondborn monozygotic twins who have the exact same genetic makeup. If we look at dizygotic twins who share only half their gene, we can just look at the shape of this distribution and see that it is much less similar. So there's still a relationship here, but it's not anything like the relationship in reading ability for firstborn and secondborn monozygotic twins. For dizygotic twins who share only half their genes, the relationship is weaker. And that's how these are called concordance studies. This is a scatter plot showing correlations, and that's how one of the ways that we determine the heritability of a trait. Is with the heritability of intelligence, and this is looking at a number of different pairs. And we start out with adoptive parent-child pairs who wouldn't really share any of their genes. And we can see that the correlation is pretty weak. It's low, the bar is short. If we have unrelated children who are raised together, like the father brings in his child and the mother brings in her child, and we can see that the correlation in their intelligence is also weak. We can look at adoptive siblings, and we see it's kind of similar. In a way, they're like unrelated children who are raised together. Even if they were raised together from birth, there's still a relatively weak correlation between their intelligence levels. Similarly, half-siblings, They both share one parent, but not the other parent, so they share 25% of their genes. Once again, a relatively weak or weak to moderate correlation. When we get into the situation where half or more of the genes are shared, the correlations get stronger. For example, parents and children share about half of their genes, and we can see that this is a pretty strong, it's still like a moderate correlation, but it's stronger than these others were. And we can maybe attribute that to differences in age and experience, because the parents, of course, are older than the children. Fraternal twins raised together have a pretty, much stronger relationship in their intelligence. They were both born into the same home and raised the same cohort and raised similarly. And so you can see a pretty, decently strong correlation. But look at it compared to identical twins who share all their genes, but who are raised apart. We can see that this is the strongest correlation of all, even though they did not share their environment. And so this is a pretty powerful suggestion that intelligence, at least the way we measure it on intelligence tests, is highly heritable. So we can think of most traits as falling along a continuum, someone who has a very low manifestation of that trait and someone who has a very high manifestation of that trait. And that the human condition, of course, if we take the person with the lowest manifestation and compare that with the person with the highest manifestation, we're going to see some big differences. So let's take a trait like shyness, and we'll just use that word shy. There are other words, but let's just take the shyest possible human and just imagine that person. They're the shyest person in the whole wide world, and that would be the lower end of the continuum. And now let's take the least shy human, the most outgoing person in the world, and that's the other end of the continuum. And let's imagine that there's some genetic contribution to this. Let's say someone's genotype for shyness falls right around here. So there's a genetic contribution. This doesn't mean that this is exactly how shy they're going to be. The phenotype for shyness, what actually manifests, first off, assuming the gene is expressed, or the combination of genes are expressed, then their phenotype could be anywhere in here. Like they could be shyer than their genotype suggests, or they could be more outgoing than their genotype suggests. And this is dependent on the environment. So things like early touch-based care, but then also what we're going to talk about now are passive, active, and evocative factors. So what we're talking about then is that there tends to be a correlation between a person's genes and their environment. And why is this present? So first off, what are we looking at? If we take a person's level of outgoing genes, their genes for outgoingness, and then we measure how outgoing their environment is, like do they live in a really socially engaging environment at school, at home, and how are they living their life, we find that there's a relationship. The more outgoing their genetic fingerprint is, the more outgoing their environment tends to be. Why is this? We have three explanations for this. So let's take a look at a couple of these explanations. We call these gene-environment correlations because, again, these two factors tend to be correlated. So first, we have a passive gene-environment correlation. This R here stands for correlation, a passive gene-environment correlation. And what that means is that the child didn't do a thing. The child was just genetically similar to their parents. They got the gene from their parents, and because their parents also tend to be either shy or outgoing, the environment in which they raise their child tends to be similar. So if we have an outgoing genotype, the parents probably also had it, and we have an outgoing phenotype. And it's just passive. The child didn't do anything to get put into the situation with this outgoing mom, but the environment is outgoing, and that's what the child becomes comfortable with. On the other hand, if we have a quiet, or we might say a shyer genotype, we tend to have a shyer phenotype. And so once again, there's a passive thing. The child is just born into a home where the parents tend to enjoy quiet activities. So that's passive because the child has not done anything. But there are also active phenomena, and what that refers to are people's choices of environments. So they choose what they want to do in their lives. That's why it's active. So for example, the active, an outgoing child, a child who is this one right here, when they go into their kindergarten classroom, they're going to choose things to do that are probably social. They're going to make different choices than a quiet child who might seek out things that tend to be more quiet. This is called active because the child has a choice. They're doing something. They're taking action. Finally, we can have an evocative gene-environment correlation in which children receive responses from others that are influenced by their genotype and interpreted differently. So it's reactive. They bring out these actions in other people. And we can imagine this outgoing child that we're following down this vertical trajectory. This outgoing child tends to elicit or bring out outgoing behavior from others. So she might walk into the room, or he might walk into the room, and just their presence make others feel more outgoing and engaging. Whatever they say and do, the energy they bring into the situation brings out outgoing behavior in other people. Conversely, this quiet child would probably tend to evoke quiet behavior in other people just by the energy or actions that they bring into a situation. So these are the three types of explanations for why there tends to be a correlation between genes and environment: passive, active, and evocative. But we now know a little bit more. We now know that epigenetics also may play a role. So we used to believe that we are who we are because of some combination of nature, meaning the genes that we were born with, the characteristics we were born with, and nurture, meaning what our environment is. We now find this to be an outmoded model. So we now know that nature versus nurture is a shallow explanation, and epigenetics is the third explanation for this. What does this mean? And I would go into a story of how I came to know of this, which relates to lead poisoning that happened in the entire water supply in Flint, Michigan, in which I heard a doctor say that the lead poisoning experienced by the babies that she was seeing in her medical practice were such that they could be passed along to grandchildren. And I didn't understand. How could something experienced by a baby change their gene or whatever it was to pass along to their grandchildren? Now you're going to watch an excellent video by Courtney Griffiths, which will provide the best explanation. But in the meantime, I'll give you some highlights before I wrap up. So this is the doctor. These were some of the babies she was seeing with lead poisoning. And this is what she said. Epigenetic research suggests that lead exposure in women can lead to DNA changes in their grandchildren. This is what epigenetics is: DNA changes in the grandchildren. And so just a little bit about what's going on here. We now know that we have the genes, the blueprint for what protein should be produced or what traits should be represented. And we have all this extra DNA that we used to think was junk DNA that we now call non-coding DNA, but it actually helps to determine how and in what situations the gene should express itself. It's adaptive based on what's happening in the environment. So, just a quick metaphor. Epigenetics are processes and mechanisms that, quote, affect the ability of the DNA, but do not change the DNA itself. Once again, they affect the activity of the DNA, but they do not change the DNA itself. So a simple metaphor: DNA is like a blueprint containing specific instructions on how to build something, like a human. RNA is like the workers that create and assemble the pieces based on the instructions. And we know that sometimes even the best blueprint can't be done just the way it's described because maybe there's a tree in the way, or maybe the ground turns out to be unstable. So we can think of epigenetics being like the construction foreman on site saying, we can't do this plan. We're going to change this instruction a little bit because that's not what the situation will allow. And so that's a little metaphorical explanation. Another metaphor, we might think of DNA as the recipe. RNA is the process of following the instructions and making the recipe, but epigenetics are like the notes in the margins that adapt the recipe to whatever's needed in the current situation or what the current preferences are. In brief, you'll get more of this in that little video, just in brief, there's like chemical tags and all that, what we call junk DNA or non-coding DNA, that are an interface between the gene and the environment. And so if lead is coming in from the environment, then there's a message to the gene saying, oh no, don't express that way. And it's done it through what we call DNA methylation that silence a gene, or histone modification. Remember, the histone is the structure part of the chromosome, the proteins around which the DNA is wrapped within the chromosome, and that also determines what genes turn on and off. So with that, I'm going to stop recording, and I hope you find this helpful and that you're able to do well on your quiz with the information provided here.